Hello all, welcome back to the BioRocks channel. Today in this video, I'll be talking about the structure of the chromosome. As I have promised you all that I'll be coming up with the series of the genetics where I'll be teaching you about the fundamentals of the genetics. So in the last one of the videos, I have spoken about the structure, or I have spoke about actually the tertiary organization of the DNA into the chromosomal structure, right? This you can see on your screen here, children. What we have spoke was the double helical DNA molecule. As you can see here, the double helical DNA molecule is going to come together as a thin chromatin structure by forming the nucleosomes with the octamer proteins, which are being wound up with negatively charged. You know that the octamer proteins are positively charged and the DNA was negatively charged. They wind around, form a nucleosome structures. And so this particular chromatin is going to actually, the nucleosomes are going to move further back into a solenoid-like structure, which is going to further condense and is going to extend into loop-like structures that you can see here, these loop-like structures. And further, there will be a condensation, which is forming the chromosomal-like structure that you can see that during the mitosis, you can find usually the structure of chromosome and in this particular form. So this is a synopsis for one of the last videos that I have made, how the DNA is being organized into the structure of the chromosome. Now, coming on to the structure of the chromosome, a chromosome usually is going to comprise of a single chromatid arm. This arm is being called as a chromatid. Usually, the single chromatid arm is being present during the G1 phase, okay, of the cell cycle. So during the G1 phase of the cell cycle, the chromosome is going to have only a single chromatid, and single chromatid meaning it is going to comprise of a single DNA molecule. Okay, it is having only a single DNA molecule. But during the end of the S phase or the synthesis phase of the cell cycle, okay, during the end of the S phase or during the G2 phase, you can see that the DNA replicates and the amount of the DNA gets duplicated and it forms two identical DNA molecules. It forms two identical DNA molecules, due to which this particular chromosome now is developing during the end of S phase or the G2 phase two chromatid arms. So these arms are nothing but the chromatids. Got it? And remember that a functional chromosome should comprise of most important parts. What are those parts here? The first thing is a functional chromosome should have a centromere, and a functional chromosome should have a telomere, and a functional chromosome should even have a origin of replication also, because you know that a chromosome is nothing but a DNA molecule. So in order to trigger or start up the initiation of the replication, we need origin of replication. If at all, if there will be a mutation in this origin of replication, there will be no further replication in the DNA molecule, which is going to lead to the fatal consequences. As you all know about it, there would be no replication of the DNA. Having a duplicated DNA, having the two chromatic arms, having two chromatic arms, with the central structure which is being called as centromere. This is being called as the centromere. Remember, children, the centromere is going to be a constricted area. Okay, this is actually a constricted area, right? So this is a constricted region that we okay, adenine and thymine. nucleotide, that is, we call it as these nitrogen bases sequences are repeating. Mostly you can see that the 90% of this particular ones in the centromere are going to be both of these area only. Okay, so the epigenetic changes that whatever you can expect can be seen in the chromatin, can be seen in the chromatin of the centromere. Okay, so these are the ones, as I have told you in the last videos, as a heterochromatin is a one which is undergoing the epigenetic changes. So here, as you have this heterochromatin, the centromere heterochromatin will be undergoing the epigenetic changes that we can see. Okay, and not only this, children, you can see another important, most important aspect of the centromere is in almost all the other chromatids, in the chromatid areas, you can see that the octamer proteins are going to have two H3 proteins, two H4 proteins, two H2A proteins, two H2B proteins, but the centromere is going to have the variant of the H3 protein, okay, variant of histone, that is H3 protein, and that variant of H3 protein found in the centromere is being called as a SAN A. SAN A we are going to call, or we call it simply it as a SAN H3. This is a variant of a H3 histone protein that is found only in the centromere area. And this particular variant of the H3 protein, which is a SAN H3, is going to actually bring about the changes in the centromere's chromatin, that this is going to actually attract the complex of proteins. This is going to attract the complex of the proteins which are being called as kinetochore. So this is the one which is attracting the kinetochore complex of proteins which are getting attached to this particular, what we call as your centromere. As you can see, children, let me show you that you can see here that this is going to be your centromere, and you can see this green color structure here present here, right? This one, let me color. So this is a what you call is a complex of proteins which are being called as kinetochores, which are attaching to the centromere chromatin structure. And these kinetochores are those complex proteins which are attracting the microtubules of the spindle fibers. These are the microtubules of the spindle fibers. These are being attracted and attached to the kinetochore. So imagine if the centromere would not have the protein called as a H3 variant, SEN H3. If this has not been present, then there would be no kinetochore and there would be no attachment of the spindle fibers to the chromosome area. Okay, and that would not lead to the division or cell division. You can't even imagine about the further proceedings of the cell division, right? So this is also another important aspect of the centromere. And besides this, children, your centromere is even going to send the inhibitory signals. This is going to send the inhibitory signals, and until the chromosomes are attaching to the spindle fibers. If there is no attachment of the spindle fibers, then your cell phases will not proceed from the metaphase to the anaphase. And those inhibitory signals are also being sent by the centromere only. Okay, so the centromere, it is sending the inhibitory signals if there is no attachment, if there is no attachment of spindle fibers to the centromere, that is to the chromosomes. So even this is also most important functionality of the centromere. So it's like having the variant of the H3, having the attachment of the kinetochore, attracting the spindle fibers, the microtubules of the spindle fibers, sending the inhibitory signals. And these are all the most important aspects of the centromere that we can find about. Okay, these are the most important things. I hope you understood about the centromere. Now coming and talking about the ends of the chromosomes, which are being called as the telomeres. As I told you that every functional chromosome should have a centromere, it should have a telomere, it should have an origin of replication. Who are those? These are the functional chromosomes. If these chromosomes would not have these would not regarded to be a functional ones, right? So what are those telomeres? Telomeres are actually nothing but the end stabilizers. These are the ones which are going to act as a cap like things which are being present at the ends of your DNA molecule, which are protecting the chromosomes from sticking together. Okay, let me show you first what are those telomeres. As you can see here, these are the ends which are acting as the cap stabilizers, or the caps which are avoiding the sticking of the what you call is chromosomal ends. And remember children, sometimes your chromosomes even may break down. Okay, when they break and they produce several segments of the chromatin, these ends of the chromosomes may stick together and they form aggregates, and these aggregates will be later on what you call is deteriorated because they become non-functional. So to avoid the aggregation of the chromosomal ends, the chromosomal ends, sticky ends are going to have these telomeres, and usually your telomeres, sorry, are going to have mostly the adenines and thymines. But you can see children on your screen that the end here at the 3' is having, you can see that the most of the guanines are repetitive. You can see that the G's are very, very repetitive. And you can see that the complementary stand is having most of the C's. This you can see even in the humans and the eukaryotes, and you can see in the plants, in the protozoans. And remember that you can see that this particular end is overhanging, or this is extending, right? This extended end of the guanines, which is being present here, is going to be called as an overhang of the G end. Okay, we are going to call this end as a G-rich single-stranded overhang. We are going to call this as this one because this is extending and it is actually hanging above the complementary C strand, right? So this is the most important thing to be remembered. And remember children, this particular overhang is going to form a long loop which is being called as a T loop, and this T loop is being associated with one of the proteins which is being called as a sheltering protein complex. These togetherly are going to actually form these cap-like structures, which is allowing the avoiding of the sticking of the ends of the chromosomes and formation of the aggregates. I hope you understood that these G strands with the complexes of the sheltering protein complexes forming this particular caps called as a telomeres, which are avoiding the sticking of these chromosomal ends. Okay, I hope children, I made it clear for you all children, the most important aspect, that is a structure of the chromosome and the functionality of the chromosome here. And remember, even in the chromosomal chromatid arms, we see the presence of the chromomeres and the chromonema. The chromonema is going to be a long thread-like structures which are being present, and which are going to be having the bead-like structures which are being called as a chromomeres. And these are the structures structures that you can see further in the chromatids. So you remember that the chromomeres are the bead-like structures on the thread-like long spindle structures which are being called as the chromonema, which are being present inside the chromatids. Okay, children, I hope this video will be helping you in understanding and revising the structure of the chromosome and understanding the tertiary organization of how a DNA molecule forms a chromosome and chromosomal structure. Okay. Okay, children, thank you all. Please try to subscribe my channel and try to share my channel with your friends, because this is going to help them all in understanding the genetics well, because I'll be releasing most of these genetic related and the DNA structure of chromosomes and most of the numericals which are coming in following up with these videos. Please do follow on and try to subscribe my channel. Thank you all. Thank you. Please do comment in the comment section. How did you like this video? Thank you all. Thank you.